successful drug development requires a number of critical components and activities to come together in a very well coordinated way to take drugs from brain to bench to clinic, as I say. And with the kind of current models where people use multiple different providers, that creates a lot of white space, so there's a significant risk in terms of time and cost. With an integrated model, where a lot of these components are under one roof, like what we have at Abzena, we can bring a lot of these kind of deliverables together and even overlap them, but also we can use real-time data to adjust and even shorten timelines. What's quite unusual about Abzena is that we have a significant front-end R&D capability, and this connects into a sort of back-end process development, scale-up and manufacturing capability. Within our R&D capability, we've got a lot of scientific and technical expertise that we use to design and develop these next generation molecules. So we've got a lot of protein engineering capability to design bispecifics, fusion proteins, bioconjugation expertise to design antibody-drug conjugates and antibody-oligo conjugates, which are very popular these days, as well as other kind of linker-payload combinations. Underneath that, we've got a great breadth and depth of analytical and bioassay capabilities that allows us to generate the data to select the best lead candidate to go forward into development. So we really develop an intimate understanding of the drugs, which pays dividends when they go forward into manufacturing and clinical evaluation. Many years ago, when I started drug development, an old mentor of mine said developing drugs was easy. You just have to answer two questions. Can we make it? And does it work? And this is at the core of developability. So we need to understand if we can actually make the molecule to the appropriate scale and the appropriate form to give to patients. But also, does it work? Does it do what we expect it to do within the appropriate safety parameters for that disease indication? So that's what we do in developability. You know, we ask a number of questions, we look at the design of the molecule, we consider where the liabilities are, we design those out and de-risk that for, you know, the more expensive stages of development in manufacturing and clinical evaluation. So developability really supports the success of the drug at the cheaper stage of development so that we can get better and more drugs into patients. When developers are selecting a cell line development CDMO partner, I think there's two key things they should be looking for. First is general table stakes, productivity, stability, quality of material. But the second is, you know, how robust is the platform? You know, is it a platform that can actually leverage to reduce the time frame from gene to IND? So, you know, we should be looking at, is the material that we produce at stable pools similar and the same as the material produced at stable clones to scale at 2,000 litres? Because that allows us then to do things like, you know, take material at stable pools and use that to take things off the critical path like analytical method development, downstream process development, bioconjugation process development, and even we're seeing more and more when people are confident in the cell line platform, using stable pool material to generate tox material. So they take the tox studies off the critical path as well. So that really helps accelerate the timeline or truncate the timeline from gene to IND and get things into patients quicker. Next generation cell line development platforms like ours, the Abzena Pro, you know, combines two key components. One is the host cell line, so we've got a robust, rapidly dividing host cell line that produces high quality material. And we also use proteomics 2G unique vector. Now this brings a lot of kind of properties to the cell line platform. So it improves stability of gene integration, it increases transcription, it improves transport and translation of the RNA. And we found this to actually be of significant benefit when we're introducing these very strange molecules, or at least the gene for these very strange molecules, which cells haven't really been used to expressing. So what we find with our next generation platform is that it's pretty agnostic to the type of molecule it's producing, and we've been seeing really high productivity levels for really funky proteins, cytokine, VHH, fusion proteins. We're getting 7-8 grams plus. So this means that, when it goes to manufacturing, it kind of unlocks now the potential of these molecules to bring better therapies to patients that need them.